My presentation is on the functional characterization of the human facilitated glucose transporter 12, also known as GLUT12, by electrophysiological methods, by Pujol Jimenez et al. Just a little bit of an introduction. Glucose transporters are membrane-bound proteins that aid in the facilitated transport of glucose into the cell from the extracellular matrix. In the human genome, there are a total of 12 GLUT molecules that are known, but research is still being done to identify more. And the majority of them are uniporters, meaning that they only transport glucose in and out of the cell and no other molecules. They transport glucose down the concentration gradient, meaning from an area of higher concentration to lower concentration, until a solution equilibrium is reached, meaning that there's an equal movement into and out of the cell of glucose. There are 3 total subclasses of glucose transporters, with each class reflecting a unique function, property, or location of the glucose transporters. The focus of this study was on GLUT12, which is a class 3 glucose transporter. And it was found to be most widely expressed in cancerous tumor cells, muscle cells, and adipose tissue in humans, and was first isolated from breast cancer cells, and was found to be a key protein in supplying energy to cancer cells that had been malignant and were continuing to grow. GLUT12 was originally thought to be an ancestor to GLUT4 due to their insulin sensitivity, but the majority of the functions of GLUT12 are unknown. But it is known that sodium and hydrogen ions do influence the uptake of glucose and transportation by GLUT12. The purpose of the study was to be able to just basically outline some of the functional properties of human GLUT12 in African clawed frog oocytes. So basically they were expressing GLUT12 in the oocytes or the underdeveloped egg cells, and they basically just wanted to be able to determine the effects that hydrogen and sodium ions had on glucose transport, and also the effects that protein kinase activators of protein kinase A and C had on glucose transport. And then they followed that up with determining the effects that these ions and protein kinase activators had on the currents that were being induced by glucose associated with GLUT12 activation. So what they did was they injected circular RNA that encoded this human GLUT12 molecule into the African clawed frog oocytes in order to be able to express the glucose transport protein. Then what they did was they incubated these oocytes with radiolabeled glucose in order to determine that glucose was in fact being taken up by this GLUT12 molecule. And then what they did was they incubated these cells with the glucose transport molecule with these different conditions of ions and kinases in order to determine the effects that they had on the glucose transport. And then they also performed this electrophysiological method of using the 2-electrode voltage clamp to identify how the ions and kinases were affecting the currents associated with activation of this glucose carrier molecule. So this figure just shows the effects that sodium and hydrogen ions had on the glucose uptake of GLUT12, and the white bars represent the non-injected oocytes, so they don't express this GLUT12 molecule. The gray bars do represent the cells that were injected with GLUT12, and what you see here is at the conditions of sodium ions at pH of 7.5 and pHs of 6, there's a dramatic increase in the glucose uptake when sodium is present. But if you look at the same results for the choline buffer, which is basically just testing the effects of hydrogen ions, you'll see that there's really no significant change in the uptake of glucose. This figure shows the effects that the kinases had on glucose uptake. So the legend is the same. It's showing the non-injected and injected oocytes. And the 8-BRCAMP is the activator molecule for protein kinase A, and the PMA is the activator molecule for protein kinase C. And what you'll see is that compared to the control, the activator for protein kinase A had a much more dramatic increase in glucose uptake than the protein kinase C activator. This figure shows the currents associated with sodium and hydrogen ions on GLUT12. And the main thing that you're going to want to look at is the areas of the black dotted lines and the gray dotted lines. The black dotted lines represent the sodium ions present when glucose is being taken up by GLUT12, and what you see is a dramatic depolarization from its initial starting point of 50 nanoamps. And the gray dotted line represents the hydrogen ion effect on the currents associated with GLUT12, and you see that it's not nearly as dramatic of a decrease in the current. The protein kinase activity effects on the currents associated with GLUT12. The left-hand figure shows the protein kinase A activator, and the right-hand figure shows the protein kinase C activator. In the left figure, you'll see in the area indicated by the smaller black dots, you'll see a much more sharply dramatic depolarization of the GLUT12 protein as opposed to the small black dot area of the protein kinase C activator molecule. So you can see the dramatic difference that protein kinase A has an effect on the current associated with GLUT12 as opposed to the protein kinase C activator. Some conclusions that they made about the transport properties of glucose for GLUT12. When extracellular sodium is present, glucose is being taken up nearly 40% more than when it is not. But when glucose is present, sodium is not being taken up by GLUT12, which shows that the activity of GLUT12 is dependent on sodium ions, but is not essential to the functioning of this GLUT12 molecule. The acidic pH does not have any effect on the glucose uptake, so it's basically just stating that GLUT12 is completely independent of hydrogen ions. But when sodium is present, hydrogen ions further decrease the uptake of glucose by GLUT12, which shows that hydrogen ions do interact with GLUT12 in some way. Protein kinase A, on the other hand, causes an increase in glucose uptake by GLUT12, but not protein kinase C, which shows that GLUT12 could be regulated by protein kinase A, but not by protein kinase C. Just in terms of the electrophysiological properties, the currents of the glucose-activated GLUT12 molecules increase nearly a quarter more when sodium is present versus when it's not present. There were still currents being produced when sodium is absent, showing that sodium is not essential for current induction. And in terms of the hydrogen ion effect on current induction, there's not really a significant change in the current. Taken together, these results show that sodium ions, but not hydrogen ions, contribute to induction of currents by GLUT12. The kinases, on the other hand, show more or less the same results as the glucose uptake, showing that protein kinase A activators do increase the currents of the GLUT12 by nearly 50%, but protein kinase C activators do not, showing that protein kinase A, but not protein kinase C, plays a role in regulating GLUT12. So just a general summary, it shows that GLUT12 is a very versatile protein. It's a symporter of sodium ions or hydrogen ions along with glucose, so not necessarily a uniporter like it was thought for glucose transport molecules in general. And it does exhibit electrogenic properties, as you saw with the currents induced by the protein kinase activators and the ions. And it also has its own unique properties and features, which show that it's not necessarily an evolutionary ancestor to the GLUT4 molecule like it was once thought. And these properties do indicate that there may be a specific